Bismillahirrahmanirrahim. Now, second video on the chapter on cell structure, and this will be the topic-wise MCQs. Question 43. A light microscope is used to observe two membranes that are 200 nanometers apart. Again, this is a question which checks your English. How far apart are the membranes when the objective lens change from low power X40 to high power X400? Well, if something is this difference apart, and you magnify it, but originally it will remain the same distance. But of course, if you zoom something, it doesn't make them different. It will remain the same, but you're just zooming it, so you're just magnifying it 40 times. Now you're magnifying it 400 times, but it doesn't change the original how far apart they were. If they were 200 nanometers apart, if this was 200 nanometers apart originally, then it will remain 200 nanometers apart. But of course, they're not saying how the image would change. They're saying how far apart are the membranes when the objective lens is changed. Well, they are actually 200 nanometers, but now because you've magnified it so many times, they are. It's a sort of a trick question. I don't like these sort of questions because I don't think they check biology. They check more your English. The electron micrograph is of a chloroplast. The length of the chloroplast along the line shown is 80 millimeter. The actual length of the chloroplast is 10 micrometer. So they've given you that it is now in the diagram, it is 80 millimeter. Now I always say is convert it into micrometer. So whatever is given to you here, the unit here, so it's 80,000. And so you divide it by 10. It's left with 8,000. So 8 into 10 raised to the power 3. So basically, you see, you convert it into the same unit, and then of course it makes it very easy. Then coming on to the next question. Now, question 45, the diagram below is drawn from an electron micrograph of an animal cell, which represents the same cell seen under a light microscope at X400 magnification. So you're not going to see any details, and this is all what you will see is C. You can't be seeing this ER, this Golgi, and all this ER and RER and mitochondria. You can't see these detailed at 400 magnification. They're these structures are not very easy to see. Light microscope is only something less than 200 nanometers you can see. It's only 400 nanometers, so you have to see something which is bigger than 200 nanometers. Now you can't see all these details. Which feature enables an organism to be identified as a prokaryote? You know, prokaryotes are bacteria. So they have a cell wall, they have circular DNA, but then it says which feature enabled to be identified as a, but cell walls are also in plants. So the only thing which is only typical to prokaryotes is circular DNA. They don't have a nucleus, they don't have ribosomes, and the cell wall is present, of course, in many others. So which feature enables an organism to be say, okay, definitely this is a prokaryote. Cell wall is present in everything. Fungus has a cell wall. Bacteria has a cell wall. Plants have a cell wall. Algae have a cell wall. 47, the diagram shows an electron micrograph of a typical animal cell. What is the function of the membrane system labeled X? Now this is RER, so it's protein synthesis and transport. If it was just, it's a membrane system. If it was just the ribosome, then it was protein synthesis. If it was SCR, this is all the SCR, then it's lipid synthesis, but lipid synthesis and transport, if I had labeled this whole thing. Then question 48, an animal cell and a plant cell are placed in distilled water. The animal cell swells and bursts while the plant cell swells but does not burst. What accounts for this difference? Animal cells have no cell wall, that is why they burst. Plant cells have a cell wall which protects them and which just prevents it from bursting. Question 49. Which eyepiece and objective lens combination enables you to see the greatest number of cells in the field of view? Greatest number of cells in the field of view. You have to understand is that if you can see this, and I can see, say, 1, 2, 3, 4, 5, 6 cells. Now, if you magnify it, if you're going to magnify it, then you're going to zoom it. You're probably just going to see these two because now you've zoomed it. Like just look at a picture of somebody and you zoom it, so you can only see certain parts of the face. If you want to just see the glasses they're wearing, or if you just want to see the eye color of the eyes, then you can only zoom that. So you're only going to see a few. So it has to be a very. I always tell you to look at a low power when there's a Cambridge slide. And I say, look at the whole of the slide and then go on to high power, because then you know what the whole slide is all about. So first look at it, but when you magnify it, well, then you'll only see less number of. So this is what you have to see. This was 50, this was 200, this was 10 into 10 is 100, and this was 400. So this was the smallest. 5 into 10 was, we would see everything in it. We would see many number of cells. So if you sort of zoomed it, then you would only see two cells or three cells, because now you had magnified it, and you would only be concentrating on that little area where you could probably just see two cells, where initially you could see six cells of a prokaryotic cell. Of course, the endoplasmic reticulum is absent, so it has to be these two, couldn't be these. So this is how you cut it out. Genetic material in a prokaryote is DNA. So the genetic material is DNA, because it's viruses which have DNA or RNA. So this is how you have to be. The answer was A. Then 51. The following are all features of eukaryotic cells. Which of these has a single membrane? Which of these has a single membrane? You know, chloroplast has a double membrane. You know, mitochondria has a double membrane. You know, nucleus has a double membrane. Golgi and the lysosome have a single membrane. Plant cell are fixed, stained, and viewed through a light microscope. What would be clearly visible at a 400 magnification? You can't see these details. Cristae of mitochondria, grana of chloroplast, ribosome. You probably, this was the largest thing that you could have seen, so it was nucleoli. Which size range would include most prokaryotic cells? So you can see here, this is the A part, then the B part, and then the C part, and then the D part. So this is all increasing. And these, of course, are nanometers. This is what the virus sizes are. So you have to know the sizes. You please remember, you have to make a rough idea of the sizes. You have to be clear about the sizes. The photomicrograph shows some plant tissue and the magnification is x200. What is the diameter of the cell marked Z? Well, you have to measure it, convert it into micrometer, and then divide by 200. Now, this is probably not the original diagram, so I can't say what was it in the original paper. Which range of sizes would include most eukaryotic cells? Again, sizes you've got to know. Again, then question 56, trick question. Why do I say it's a trick question? Because you don't read it very carefully. A student was asked to draw a planned drawing of the plant tissue shown in the photomicrograph and to annotate two observable features. Observable features we didn't read. You don't read observe. What can you observe? The person has a lung infection. How can you observe that he has a lung infection? You can observe that he has a cough, maybe, but you can't observe that haven't gone. You've got X-ray eyes and you can see what is happening in his lungs. So observable features, what are the correct annotations? Annotations means a slight description which you give. This is called an annotated diagram. Please remember that. What are the correct annotations? Epidermis, darkly stained layer of cells, xylem, hollow vessels. Now, why could you not have said B? Form single layer of cells, xylem strengthened by lignin. Where did you see the lignin? I can't see the lignin in it. And you can see the lignin in it. Phloem, small cells, xylem, empty cells to transport water. Well, who knows what is being transported where? We can't see it, we can't observe it. Vascular bundles arranged in a regular pattern, xylem, large dead cells. I can't see any dead or living things here. So please remember, it has to be observable features. Observe means something which you can observe. Which structures are present in all eukaryotic cells but not present in prokaryotic cells? Present in all eukaryotic cells but not present in prokaryotes. So chromatin is that DNA which is wound around the histone protein. What restricts the resolution of the light microscope? The wavelength of visible light. You know, it's half the wavelength. So anything which is, so that is why light has 400. So the wavelength of light is the restricting the resolution. If there's any ambiguity, you've got the year and the number of the question, and you can easily find out. If you are not very clear, you can look up the exam report or the mark scheme of that year. Now, question 59. The diagram shows an electron micrograph of virus particles in a human nucleus. Virus particles inside a human nucleus. What is the diameter of the labeled virus particle? And you have to figure that out. You have to measure it and all that. What is the diameter of a typical plant cell? Again, this is something which you must know. Where would cisternae be found inside a cell? Cisternae are found in the endoplasmic reticulum and the Golgi. Please remember there's something known as cisternae and there's something known as cristae. Cristae is in the mitochondria. Ribosomes exist as separate subunits that bind together during protein synthesis, and they're made of ribosomal RNA and protein. You might not have studied this as yet. 63. Which cell components contain mRNA? You see, wherever there's DNA and ribosomes, there'll be mRNA. So chloroplast has it, mitochondria has it, nucleus has it, and rough endoplasmic reticulum, of course, will have the mRNA attached to it. But it might not be easy at the moment because if you're just done your A1 and you've just done chapter 1. Now, this again is a question which is often asked by all of you. The diagram shows a stage micrometer and small division. So this is, let me give it another color. This is 0.1 millimeter, right? So 0.1 millimeter. This is 0.1 millimeter. It is viewed through an eyepiece containing a graticule. Now this is the eyepiece is the graticule. This whole thingy is the graticule. And it's got 100 divisions. And I told you eyepiece graticules have no units in them. To question number 64. I think I've devised a new strategy of how to do this. My maths is not as good as my biology, but I'll try my best. The diagram shows a stage micrometer in which the smallest division are 0.1. So as I've shown you, this is 0.1. This dark line is 0.1 from here to here. Now this 0.1 is equal to 40. So first of all, let's figure out this in micrometer. So let's convert the 0.1 millimeter. This point is equal to 100 micrometer. So we fix this out in the beginning and everything becomes easy then. So 40 is equal to 100 micrometer because we know the answer is in micrometer. So 1 is equal to 100 over 40. So 4 is how many? Why do we do 4? Because the stage micrometer is replaced by a slide of a plant cell. What is the width of a chloroplast? Now the chloroplasts are these structures. I'm going to just rub this off and then I'm going to show you the chloroplast. The chloroplasts are these structures. Please remember, this is a chloroplast, this is a chloroplast, this is a chloroplast. This is, please remember, this is the nucleus. So these are the chloroplasts, and roughly, I mean, if you look at it, I mean, I'm drawing a line, but this is not very clear. So it's about 4 divisions. So this is why I've used these 4 divisions. So 140 is equal to 100 micrometer. 40 is equal to 100 micrometer. Yes. 0.1 is equal to 100 micrometer. So 40 is equal to 100 micrometer. 1 is 100 over 40. 4 is whatever. And the answer is 10 micrometer. Right? So you knew this that 40 was 0.1 millimeter. So let's convert that millimeter into micrometer. So 40 is equal to 100 micrometers. So 4 kitne honge? Bas, so it's easy. 4 will be how many? So you just do the simple maths. 40 is equal to 100, 1 is equal to so much, and 4 is equal to so much. So yeah, this green thingies, these green were the ones which I'm trying to make for you. These are the chloroplasts. This is the chloroplast. These are so roughly you would say this, and this of course this large thingy, this large blob here was the nucleus, and this is what you would have confused. That's why they give you all these different options. Then question 65, which processes occur in you and prokaryotes? Hydrolysis, mitosis. Mitosis never occurs in prokaryotes. Bacteria divide by binary fission. Prokaryotes divide by binary fission, and that was a clue that if you didn't know this, well, you're going to get this wrong then. So transcription, you haven't done. Translation, that is of course what you had to understand. It was 1, 3, and 4. Using a stage micrometer scale, 1 unit of an eyepiece graticule was calculated about 0.005 millimeter. The diameter of a spongy mesophyll was counted as 3.5 units on the eyepiece graticule. What is the estimate of the diameter cell? The simple maths. Membranous sacs containing products of metabolism formed by the endoplasmic reticulum in cells. What are these products? Where are these products used? So of course they will be used inside and outside the cell. Whisking tubing is an artificially partially permeable used to demonstrate diffusion. Glucose molecule can pass through the pores in the membrane, which are approximately 2.4 nanometers in diameter. Which of the following could pass through the pores? Bacteria, no. Hemoglobin, no. Ribosomes, no. Glycogen, no. Which of the structures are found in photosynthetic prokaryotes? Now, of course, the child would have thought of chloroplast, but please remember they do not have membrane-bound organelles, so 5 was wrong. So this was wrong and this was wrong. Chloroplast cannot be present inside prokaryotes. And of course, what was 2? Cellulose cell wall was also not present in prokaryotes. So this was wrong as well. So that's why the answer was 1, 3, and 4. Then coming to the next question is 70. A human aorta has a lumen width of 2 centimeter. A human red blood cell has a diameter of 7 micrometer. How many red blood cells could be laid end to end across the diameter of the aorta lumen? So of course you have to do a maths, and so you would do that. Which steps are needed to find the actual width of xylem vessels? Viewed in transverse section using X40 objective lens. Actual width of the xylem vessel. So convert from millimeter multiplied by 10 is about 3. Calibrate the eyepiece. Use stage micrometer on 10 objective lens. That's rubbish because we were using a 4 objective lens. So 2 is definitely out. Now that was very easy. If you could know 2 was out, so 2 was here also, 2 was here also, and 2 was here also. So A, B, C are all wrong. And that left you only with D. That's called smart work. That's called smart thinking. 72. Specimen is viewed under a microscope using green light with a wavelength of 510 nanometer. If the same specimen under the same condition but using red light with a wavelength of 650 nanometer, what effect will this have on the magnification and the resolution? I've done this in a previous question. Will remain the same and decreased. Which cell organelle does not contain nucleic acid? Chloroplasts have DNA, lysosomes have some enzymes in it, so it has got some nucleic acid, and sometimes there is some ribosome, of course, have RNA, so it's the Golgi. This is a little controversial, this question, because some lysosomes have some nucleic acid, so that is why this is a little confusing, and I would have also got this wrong. 74. The diameter of living cells vary considerably. The typical diameters are a eukaryote, such as a white blood cell, 1.5 into 10 is per 1 micrometer, prokaryote 7.5 into 10 is per 2 nanometer. Use these measurements to find the maximum number of each cell type which would fit along a line 1 centimeter long. Basic maths. But if you struggle with maths, then I'm sorry, you'll have to improve your maths. Whenever you have this problem, what you have to do is white blood cells were in micrometers. So 1 centimeter is equal to 1000 micrometer. And 1 centimeter is equal to 3 zeros of micro and 3 zeros of nanometer. If you get these right, then you get the whole thing right. So be systematic in it and do it in a profound manner, and I'm sure you'll get it right. 75. Cells consist of a number of different components. Which row shows the components present in both prokaryotes and eukaryotes? So lysosome जिसमें भी था, that was wrong. So we know B and D was wrong. Lysosomes are not present in prokaryotes, so they couldn't have been in both. Golgi is not present in both. Ribosomes are present in both, so that was correct. And cell surface membrane is present in both. Then the diagram electron micrograph an animal cell which represents under a light microscope at 4. I told you this previous question. You don't see any details under the light microscope, so the answer was C. Then coming to the next question, again is one of those favorite questions which you all dislike so much. The diagram shows a stage micrometer with divisions 0.1 millimeter apart, viewed through an eyepiece containing a graticule. So again, this is 0.1 millimeter, which is equal to 100 micrometer. So 100 micrometer is equal to 40 divisions, so 50 to 90. Plus, please see, they might change it in some paper and you'll stick to the 40 mark, and that will make you do a big mistake. What is the area of the field of view? And of course, know that pi r square. So if the radius, the whole thing was if 40 is equal to 100 micrometer, then what was 1 is 100 over 40, and then what was the whole thing? The whole thing was 100, 0 to 100. So 100 is equal to what? And this is how you are figuring it out, and that would get you the radius, and then you would get the half of that. You can see the working, I've done that. So 40 is equal to 100 micrometer. Now because it's simple, 1 will be 100 over 40, 100 will be 100 over 40 into 100, and that's 250 micrometer. This would be the radius, this would be the diameter, so the radius would be 125 into 125. Pi r square, pi r square. So radius twice, and that would be the answer. Then coming on to 78, which observations suggest that a cell is eukaryotic? Cytoplasm includes endoplasmic reticulum. Protein molecules are associated with DNA. Ribosomes distributed. So this was part. The answer was D. Which structures are found in both animal and plant cells? Animal and plants is centriole. So centriole is not present. So the ones which have one, jidhar bhi hai, wherever there is one, that is wrong. So this is wrong and this is wrong, because centriole is present only in animal cells. So then you narrow down to B and C, 2 and 4 or 2, 3 and 4. So both animal and plant cells, again this is an chapter 1 question, differences between plant and animal cells. 80, which structures are measured using these units? Chloroplast, ribosome, nucleus, 10 raised to power minus 3, 10 raised to power minus 6, 10 raised to power minus 9. And the only answer was xylem vessels, nucleus and ribosome. You have to know these, you have to know, and you have to become slowly, you've got to become very comfortable with this. I think we're now coming to the last two questions. 81. When making measurements in experiments, which methods have parallax errors? Using a calibrated eyepiece graticule to measure length? No. Using a measuring cylinder and using a ruler. Parallax error is when you're looking at it and you're looking at, so please put up in the Google search, look up parallax error and understand it if you don't know what parallax error is. Membranes within and at the surface of cells have different roles. The diagram allows the identification of the various organelles within the cell by describing the membrane structure and function. So membrane, cell membrane encloses a structure within the cell. No pores present. Possess pores. Allows exit of mRNA, nuclear pore, protein synthesis, RER. No pores present. Single membrane. Transports enzyme, lipid synthesis, SCR. Transports enzymes, lysosomes, double membranes. Allows oxygen to enter, mitochondria, allows carbon dioxide to enter, chloroplast. Which of the outcomes shown below correctly identifies the organelles that possess the membrane and function concerned? So here we have the answer to it was, one was nucleus, two was rough ER, and this is the answer was this, vesicle, smooth, mitochondria, chloroplast. Okay, I'll try to fit in the whole question so that you all can really understand it. So this was the question. I'm sorry I can't fit in the whole question in one page. And so just pause it if you have to read the question and then look at the answer. With this we come to an end to the second video on the MCQs on the first chapter, which is the cell structure, and I will inshallah very soon doing the second chapter as well. Best of luck and thank you for watching.